Hello everyone, my name is Awad Walied. I'm a second year med student and today I'll be giving you a very important PAL inshallah. Today's PAL is MOL Pre-Mid and we're gonna cover the high yield concepts. And I've also tried my best to cover everything, but I ended up with covering 80 to 90% of everything. If this number is good with you, then this PAL is enough for pre-midterm inshallah. If not, then I recommend you go through the individual PALs or the lectures itself. This, I believe 80 to 90% is a good number. And for cell signaling, I aimed for 95% because cell signaling is easier to cover. Now, with that being said, I want you guys to know the format of the entire lecture, and it's pretty unique, so please focus. Firstly, we're going to cover cell signaling 1, 2, and 3, and it's going to be question based. Now, I know this is a recording, but I still want you to pause, try to actually answer, because this is a review. You're expected to know. And if you don't get it correct, we're still going to go through it, okay? And then we have molecular techniques, enzymology, and all the Dr. Hana lectures at the end. The remaining three parts are going to be normal teaching, and we're going to have a few questions here and there. So stay attentive. Now, let's begin. I just want to show you guys the format of the questions. So I'm going to show you a question. I'll just read the question. You guys should pause and try to answer it. And then I'll explain. And then we're going to go back and I'll explain each individual option and make sure you understand the question fully. So let's begin with question 1 of cell signaling 1. Which of the following is correct about endocrine signaling? 3, 2, 1. Okay, let's begin. So the first form of chemical signaling is autocrine, and in autocrine signaling, the cell targets itself. Very simple. Auto means self, and so it targets itself. The second one is signaling across gap junctions, and over here the cell targets another cell which is connected by gap junctions. And I want you to think of this logically. If it's connected by gap junctions, it means they're literally touching each other. Okay? So you cannot target a distant cell by gap junctions. Doesn't make any sense. And we have one of the options like that. That's why I'm explaining it. The third one is paracrine signaling, and it's when a cell targets a nearby cell. However, they're not necessarily touching, okay? Through the ECF or whatever. It's just a nearby cell and it's targeting it. And lastly, we have endocrine signaling, and it's when a cell targets a distant cell through the bloodstream. And from the name, you can kind of get that. Endo means within or inside, and this means inside the bloodstream, okay? It's very important to understand and remember endocrine signaling, okay? Now let's solve the question. Which of the following is correct about endocrine signaling? A, the signaling cell targets itself. We said this is autocrine signaling, not endocrine. B, the signaling cell targets a distant cell through the lymphatic system. We did not mention anything about lymphatic system, so it's not correct. C, the signaling cell targets a distant cell through the bloodstream. This is the correct option. And D, the signaling cell targets a nearby cell connected by gap junctions. This is not correct. This is signaling across gap junctions. And E, the signaling cell targets a distant cell connected by gap junctions, which does not make any sense. This is how the entire cell signaling part is going to be. Very simple and very nice. Question 2. Which of the following is correct about membrane signaling? Okay, 3, 2, 1. Let's begin. So this is a very nice table which compares membrane receptors to nuclear receptors and the features, okay? So ligand concentration. For membrane receptors, you have low ligand concentration. You just have to memorize it. For nuclear receptors, you have high ligand concentration. And then based on this, we can predict the second line. Signal amplification is going to be present for membrane receptors because you have low ligand concentration. If you have a low ligand concentration, you want to amplify the signal. And if you have a high ligand concentration, you don't want to amplify the signal. Okay? Now, for target cell response, you have multiple target cell responses with membrane receptors. However, with nuclear receptors, you only have gene expression. Very important and high yield. Now, for the ligand type, for membrane receptors, you have hydrophilic ligands and hydrophobic ligands for nuclear receptors. And ligand examples, you have extracellular ligands, which include peptide hormones such as insulin, neurotransmitters such as dopamine, cytokines such as interleukins, growth factors such as epidermal growth factor, amino acids such as glutamate. And for nuclear receptors, you have intracellular ligands such as steroid hormones, cortisol, thyroid hormones, thyroxine, retinoids such as retinoic acid, fatty acid derivatives such as prostaglandins, and finally amino acids such as thyroxine. Honestly, you just have to memorize it. I can't do much here. So let's solve the question. Which of the following is correct about membrane signaling? A, the ligand concentration is high, is incorrect because we said for membrane signaling the ligand concentration is low. And then, okay, let's look at option B. Signal amplification is required. This is the correct option because we said low ligand concentration and then signal amplification is required. Let's look at it over here. Low ligand concentration, signal amplification is required. And option C, the ligand type is hydrophobic. We said it's hydrophilic. Hydrophobic is for nuclear receptors. The initiation signaling ligand is found in the intracellular compartment. Now we said that it's membrane receptors and we said that the ligand type is hydrophilic. And we also mentioned the examples, extracellular ligands. So it's incorrect. This would be correct for nuclear receptors. And finally, the steroid hormone cortisol, this should tell you everything, is an example of a membrane receptor ligand. Incorrect. It's an example of a nuclear receptor ligand. Okay, now question for you guys. One of them is intracellular signaling, the other one is extracellular signaling. Which is which? I'll give you 3 seconds. 3, 2, 1. This is extracellular. As we can see over here, the ligand binds in the extracellular compartment. Okay, and then it triggers a signaling cascade, as we can see. First amplification, second amplification, third amplification. So this is membrane receptor, extracellular ligand, extracellular everything, okay? And if this is extracellular, this would be intracellular. As we can see, the ligand goes through the plasma membrane, finds its receptor in the cytoplasm, and it then binds to a DNA. And we can kind of predict from this diagram is that it's affecting gene transcription, gene expression. Okay. Now, question 3. Which of the following is correct about G protein coupled receptors? Again, try to solve it. Then we're going to go through the explanation. 3, 2, 1. Oh, there's one thing I forgot to mention, guys, is that the answers of all of the questions are going to be found at the end of the pad. Okay, I have one slide with the answer bank. All of the answers are going to be there. Or if you download the notes version of the Canva document, you're going to find the answers under these slides. So let's explain. This is, 1 second, this is an example of a G protein coupled receptor. It's a receptor which is coupled with a G protein. Okay? And let's try to dissect the structure bit by bit because it's important to know every single tiny detail. Firstly, we have the N-terminal in the extracellular compartment outside the cell. And over here, we can see that the ligand binds. So ligand binds to the N-terminal, which is outside the cell, okay? Extracellular compartment. And then in the C-terminal or C-terminus, we have the G protein itself, which is the G alpha, G beta, and G gamma. And this is found in the intracellular compartment, okay? So N-terminus outside, ligand. C-terminus inside, G protein or heterotrimeric G protein. And we can also see there's 3 loops outside and 3 loops inside. These are loops which are strengthened with disulfide bonds. Important to remember, 3 intracellular loops, 3 extracellular loops. And we have 1, 2, 3, 4, 5, 6, 7 total transmembrane alpha helices. So from this, you can remember N-terminus outside, C-terminus inside, ligand to N-terminus, G protein to C-terminus, 3 loops inside, 3 loops outside, both connected by disulfide bonds, and 7 transmembrane alpha helices. And from this, if you can read it, I just wanted you to remember that G proteins are the most abundant membrane receptors. This could be a question. Okay. Now, let's try to solve this. Which of the following is correct about G protein coupled receptors? The receptor is found in prokaryotes and eukaryotes. I did not mention this, but the receptor is only found in eukaryotes, okay? So A is wrong. B, the receptor is made of 8 transmembrane alpha helices. Incorrect, we said 7. C, the C-terminal domain binds to the ligand. Incorrect, we said the C-terminal domain binds to the heterotrimeric G protein complex. And so C is incorrect. D, the N-terminal domain binds to the heterotrimeric G protein complex. Incorrect, we said the N-terminal domain binds to the ligand. And then E has to be correct, but let's just read it. The receptor has 3 intracellular loops connected by disulfide bonds, which checks out. One more time, look at the structure. Let's go to question 4. And we have 8 questions for cell signaling 1, 4 questions for 2, and 8 questions for 3. Okay. Which of the following is correct about the heterotrimeric G protein complex? 3, 2, 1. Okay. So we have 2 types of G proteins. The first one is the monomeric or RAS G protein, and the second one is the large or heterotrimeric G protein complex. The first one, we can see we have one monomer or one subunit, which is the RAS subunit, and it's bound to a GDP. In the second one, we can see we have 3 subunits, G alpha, G beta, and G gamma, and the G alpha is bound to the GDP. Now, from this, I want you to remember that RAS resembles the G alpha, or the G alpha resembles RAS, okay? And one thing about them is that they're both GTpases. So they're going to convert GTP to GDP, okay? Am I missing anything? No, we're good. Now let's look at what happens when the complex is inactive and it then gets activated. The changes, okay? So over here we have the inactive complex, and over here we have the activated complex. And this changes the moment we get a ligand binding to the, is it N-terminus or C-terminus? I know you got this correct, it's the N-terminus. So the ligand is going to bind outside. This is going to activate the entire complex. Now, the G alpha, G beta, and G gamma are all going to dissociate from the receptor. And then from that, the G beta and the G gamma are going to dimerize and go on their own. Okay? And one thing we can see, which is different, is that the G alpha is now bound to a GTP, not a GDP. And what happened is we were over here in the off state. The GTPase is the G alpha bound to a GDP. And when the ligand binds, something known as GEF, okay, guanine exchange factor, comes and it changes the GDP with a GTP. So we can see that now a GTPase has a GTP and we removed a GDP. And now this is activated. This G alpha with GTP is in the active state. It's gonna go and phosphorylate enzymes. It's gonna have its function, okay? And the G beta and G gamma are dimerized. They're gonna go on their own and do whatever. We don't care. Now, too much activation is wrong. And it's gonna, again, if you drink too much water, you're gonna die. If you don't drink water, you're gonna die. Too much from anything is wrong. And the same way we activated the complex, we need to have a way to deactivate or to stop it. And this is where GAP comes in. GAP stands for GTPase activating factor, and it's gonna activate the GTPase. It's gonna tell it to break down the GTP and chill a bit. So the GTP is gonna change into GDP and the phosphate, and the complex is gonna be in the off state or the inactive state. Now, this was a bit too much. Quick review. Ligand binds, we activate the complex. G alpha is now with a GTP instead of a GDP. The G beta and G gamma dimerize and go on their own. What happens to the GTP in details is that we have GDP with a GTPase. GTPase is a G alpha. And then Jeff changes it to a GTP. And then to stop it, GAP activates GTPase. GTPase breaks down GTP. And now we end up with GTPase with a GDP. we're back to the start, okay? Let's solve the question now. Which of the following is correct about heterotimeric G protein complex? A, it is made of G alpha, G beta, and GE. Incorrect. It's made of G alpha, G beta, and G gamma, not GE. Excuse me. B, GTP is bound to G alpha only. This is correct. It's not bound to G beta, it's not bound to G gamma. C, GDP is bound to G beta subunit only. Incorrect. GDP and GTP, whenever they're bound, they're always bound to G alpha. Okay? Now, during GDP, inactive configuration, G alpha, G beta, and G gamma are not bound together. Incorrect. We said in the active configuration they are bound together. When the complex is activated, they separate. However, G beta and G gamma dimerize and go on their own. Now, during GTP, the active configuration, G alpha and G beta separate from the complex and form a dimer. Incorrect. It's not G alpha and G beta, it's G beta and G gamma. And that's the trick. Now, pretty simple. Let's solve question 5. Which of the following G alpha subunits is responsible for activating adenylyl cyclase? Okay, 3, 2, 1. Let's proceed. This is the G protein alpha family, and the four of them which you need to know are GQ, GS, GI, and G1213. Now, for GI, I want you to ignore this. Just focus on this, okay? So we're starting with GQ. GQ activates something known as phospholipase C beta, and phospholipase C beta is gonna cleave something known as PIP2. We're gonna look into the details, which increases DAG and IP3, and we also increase the calcium. Okay, just remember the keywords calcium, IP3, DAG, phospholipase C beta, they're all related to GQ. GS is pretty simple. It activates adenylyl cyclase, which increases cyclic AMP. Then we have the opposite of that, which is GI. And GI inhibits adenylyl cyclase, and therefore it's gonna decrease the cyclic AMP. And finally, we have G1213, and G1213 activates rho kinase and guanylate cyclase, which increases cyclic GMP. You can read on it again, pretty simple, but we're gonna go into the details now. Let's start talking about G alpha S specifically. So over here we have the GPCR, pretty simple. The G alpha S is found over here, gets activated, goes on its own, and then it activates AC, which as we said is adenylyl cyclase. AC is then gonna change ATP to cyclic AMP. Very important key reaction. Cyclic AMP is gonna go and activate protein kinase A, and protein kinase A is gonna go do its function. We don't care about that. What else are we supposed to say? Okay, now as we said, too much activation is wrong and it's harmful for us actually. So we have to some way or another remove the cyclic AMP. And for that we have this enzyme known as phosphodiesterase or PDE, and it's gonna change the cyclic AMP to 5' AMP. So quick revision, G alpha S activates AC, AC converts ATP to cyclic AMP activates protein kinase A. And then cyclic AMP is converted back to 5' AMP to stop the reaction by PDE. And what about G alpha I? G alpha I is going to do the opposite. It's just going to inhibit AC. So if it inhibits AC, we have less ATP being converted to cyclic AMP, and we have less cyclic AMP, so we have less activation of protein kinase A. Pretty simple, straightforward. This is the easiest one, G alpha S and I. Now let's talk about G alpha Q. Again, it's simple, but you need to focus. So we have the GPCR over here, G protein over here, this G alpha Q. Once it's activated, okay, actually focus. Once it's activated, it activates PLC beta or PLC, phospholipase C. And this PLC is going to go to the membrane, find the protein known as PIP2, cut it in half into two parts. One part is the IP3, and the other part is the DAG. Okay? Now this DAG is gonna go and directly activate protein kinase C. The IP3 is gonna take a different route. It's gonna go to the endoplasmic reticulum. It's gonna result in the release of calcium, and then this calcium is gonna activate the protein kinase C. So if we were to ask what activates protein kinase C directly, it's gonna be DAG and calcium, not IP3. Okay? And then protein kinase C is gonna do its function. We don't really care about that. Now for the last one, which is not really G alpha 12 or 13, but this is what the doctor went through because it has a very similar function. Over here we have something known as soluble guanylate cyclase or soluble GC, okay, SGC. And what it does is it converts GTP to cyclic GMP. This is, again, if you remember, very similar to adenylyl cyclase. However, it's for GTP, not ATP. So in the beginning, you have nitric oxide. It activates soluble guanylate cyclase, which converts GTP to cyclic GMP. And if we have too much, then cyclic GMP is going to activate protein kinase G. But let's go back. If you have too much cyclic GMP, phosphodiesterase is going to kick in and it's going to convert cyclic GMP to 5' GMP. Just as we said, phosphodiesterase converts cyclic AMP to 5' AMP in the previous example. Now let's focus on protein kinase G. Once it's activated by the cyclic GMP, protein kinase G is going to have many different functions. It's important you have to read them and memorize them, such as smooth motor relaxation, platelet inhibition, and changes in gene expression. Okay, I believe that's it. Okay, let's go solve the question. Which of the following G alpha subunits is responsible for activating adenylyl cyclase? G alpha S, G alpha I, G alpha Q, G alpha 12, 13, G alpha X. Okay, the answer is clearly G alpha S, but I want to give you more questions. So we said adenylyl cyclase is G alpha S. What if it was inhibiting adenylyl cyclase? It's going to be G alpha I. Okay, what if it was related to smooth muscle relaxation, for example? It's going to be option D, which is G alpha 12, 13. And finally, G alpha Q is going to be anything about calcium, PIP2, IP3, DAG, whatever. Okay. Question 6. Which of the following signaling cascades is correct? Okay, I hope you answered that. Let's go through the explanation. Oh yeah, you guys should know this already. I did explain it in the previous one. So let's just answer it directly. Which of the following signaling cascades is correct? A. GTPase activating protein, or GAP, converts GDP to GTP. Now, is this correct? No, it's not, because we said GAP converts GTP to GDP, not GDP to GTP. What else? B. AC converts GMP to cyclic GMP. Incorrect. This is a function of GC, not AC. And then it doesn't even convert GMP, it converts GTP. Okay. C, soluble guanylate cyclase converts ATP to cyclic AMP. Incorrect. That's a function of AC. So A, B, and C are all incorrect. Phospholipase C, or PLC, converts IP3 to DAG and PIP2. Again, this is incorrect. Phospholipase C converts PIP2 to IP3 and DAG. Okay? So if this comes here, it's going to be correct. And then the last one has to be correct, which is phosphodiesterase converts cyclic AMP to 5' AMP. Again, checks out. There's nothing wrong. Question 7. Which of the following two components are required for enhanced activation of protein kinase C? We did mention this. You should go. All the options are wrong except it's this one. It's D. The correct answer is D. Because we said, is there no DAG and okay, there is no DAG and IP3 option. However, if there was a DAG and IP3 option, it would still be incorrect. It would still be incorrect because Only calcium and DAG directly activate protein kinase C. Question 8 and the last one for cell signaling 1. Which of the following protein kinases is directly responsible for inhibition of platelets? Okay, 3, 2, 1. The answer is protein kinase G. And this is it for cell signaling 1. We have 4 questions only for cell signaling 2. So let's go through them quickly. Question 1. Which of the following receptor tyrosine kinases, RTKs, consists of 2 chains linked together even before activation? Okay, you should know this, but we're going to go through it. 3, 2, 1. TKRs, we have a couple of them over here. And honestly, the only thing you should know is that they dimerize when a substrate is bound to them. When a substrate is not bound to them, they're separate, okay? There's 2 exceptions, and the doctor likes to ask about them. The first one is insulin receptor. It's always in this dimerized form. And the second one is IGF-1 receptor. Okay? With that knowledge, let's go and solve the question. Which 2 chains are linked together? It's insulin. If insulin was not here, it would be IGF-1. If IGF was present here, okay? Question 2. Which of the following domains of SRC kinase is responsible for binding to phosphorylated tyrosine residues? Okay, 3, 2, 1. Honestly, not much I can do. You just have to memorize this word for word, literally, because they can ask you about any single tiny detail over here. The first domain over here is the SH1 domain, and it's the catalytic domain, and it adds a phosphate from ATP. The second one binds to phosphotyrosine residues, SH2. The third one, protein-protein interactions with proline-rich sequences, SH3. And then we have SH4, which is unique. The exact function is not known. And the N-terminal binds to the plasma membrane. So let's go back. SH1, we said SH1 is the catalytic domain, so it's incorrect. SH2 is what? Phosphorylated tyrosine residues. So it is correct. It binds to phosphotyrosine residues. The answer is B. And then SH3, protein-protein interactions, incorrect. SH4, unique, incorrect. SH5, we don't have an SH5. If it was the N-terminal option, we would say it binds to the plasma membrane. Okay. Question 3. Which of the following proteins is responsible for converting GDP to GTP during RAS signaling? 3, 2, 1, let's solve. So what is RAS signaling? This entire thing is RAS signaling, and we only need to know a few tiny details. So we said in the beginning that RAS resembles GTP. RAS is a GTP, it resembles G alpha. Again, remember that, okay? So the first step over here is we have something known as GRB2, and it binds to the tyrosine kinase receptor in the SH2 domain. And then SOS binds to this GRB2, okay? Now, SOS is going to go to the RAS, the inactivated RAS, so the RAS which is bound to the GDP, and it's going to change this GDP to a GTP. Now recall the similar function which we saw before. So SOS is doing a similar, very similar, it's the exact function actually of GEF, the guanine exchange factor. Now this activated RAS is going to phosphorylate the RAF or promote phosphorylation of the RAF. Now phosphorylated RAF is going to do the same for MEK. MEK is going to do the same for ERK, and ERK is going to have its function, okay? So ERK is, okay, I'm not going to leak the question. Which of the following proteins is responsible for converting GDP to GTP during RAS signaling? The answer is SOS. The remaining ones they have different functions, so I don't need to go through them. Which of the following proteins is considered the most downstream in the process of signaling cascades? I did explain this over here, so no more explanations. Let's just solve it. Is it RAS? No, it's not. RAS is one of the higher ones. MEC, no. MEC is almost there, but the answer is ERK, if I'm not mistaken. Yes, it is. The answer is ERK, and RAF, AKT is, it's all incorrect. As you can see, GRB2, SOS, RAS, RAF, MEC, ERK, and that is it. This is cell signaling 2, very quick, very easy. Now for cell signaling 3, we have 8 questions. Which of the following is a peptide hormone? A, B, C, D, E. Okay, 3, 2, 1. Let's explain. We have 5 classes of hormones which you should know about, and they are steroids, peptide hormones, gaseous hormones, fatty acid derivatives, or amino acid derivatives. Honestly, I recommend memorizing all of the different options, but if you really, really don't want to, then just memorize the first one, which is cortisol, insulin, nitric oxide, prostaglandins, and epinephrine. So, which of those is a peptide hormone? The answer is insulin. Okay, insulin is not here. The answer is B, growth hormone. Okay. Um, what else? So cortisol would not be a peptide hormone. Cortisol would be a steroid. And then dopamine, thromboxane A2, and hydrogen sulfide. Dopamine is an amino acid derivative. Thromboxane A2 is a thromboxane under fatty acid derivatives. And hydrogen sulfide is a gaseous hormone. Question 2. Which of the following is a glucocorticoid steroid hormone? This is ERP knowledge, kinda. You should still know it. So 3, 2, 1. Let's go through the types of steroid hormones. So we had 5 different classes of hormones, and now we're gonna dig deep into this class. So we have 2 different direct classifications. The first one is the corticoids. Second one is the gonadal hormones. Under the corticoids, we have glucocorticoids, which is cortisol, and mineralocorticoids, which is aldosterone. The way I remember aldosterone is mineral, water, and then aldosterone has this function, okay? So if you remember aldosterone for mineralocorticoids, immediately just link cortisol with glucocorticoids. I don't know if I'm making sense. I hope I'm doing. Now, these are the 2 examples you have to remember only for corticoids. For gonadal hormones, you have male or the androgens and the female sex hormones. The androgens, you have testosterone and DHT. And then for the female sex hormones, you have the estrogens, which are estradiol and estrone, and the progestogens, which are progesterone. With that knowledge, let's solve the question. Which of the following is a glucocorticoid steroid hormone? The answer is D. Okay, the answer is cortisol. Testosterone is an androgen, estradiol is a estrogen, and then aldosterone is a mineralocorticoid, progesterone is a progestogen, whatever that is. Question 3. Which of the following is correct about a typical steroid receptor, structure, and function? Okay, 3, 2, 1. Let's dissect the typical steroid receptor structure. So in the beginning, closest to the N-terminus, we have the variable domain, which interacts with other transcription factors. And then we have the DNA binding domain, which the keyword you should remember is zinc finger motifs. You don't need to understand it. And then next to that, we have the domain for dimerization. It's where 2 receptors join. And then we have the hormone binding domain, which is the site where the ligand binds. And finally, the NLS, and this is the 5th domain. And the NLS is simply a signal. I think you might remember this from MOL 1. It's a signal which tells this entire protein to go to the nucleus, okay? With that knowledge, let's solve the question. So which of the following is correct about typical steroid receptor structure and function? The variable domain is the closest domain to the N-terminus. Let's look at that. Variable domain, first one, closest to N-terminus. So the answer is A. Now, the DNA binding domain is the closest domain to the C-terminus. Incorrect. DNA binding domain is this domain, and it's far away from being the closest to the C-terminus. The dimerization domain is characterized by zinc fingers. Domain for dimerization, it's not. The DNA binding domain is characterized by zinc fingers. And then the NLS is responsible for receptor trafficking to the cytoplasm. Incorrect. We said it's responsible for receptor trafficking to the nucleus. And then E, the ligand binding domain, is responsible for receptor dimerization. And this is incorrect because the domain for dimerization is responsible for that. Okay. Question 4. Which of the following is correct about a motif? 3, 2, 1. Okay, so we have a big table, has everything we need to know, motif and domain. Motifs are super secondary in structure. Domains have a tertiary structure. Motifs are formed by connected alpha helices and beta sheets through loops. Domains are formed by disulfide bonds, ionic bonds, and hydrogen bonds. Motifs have structural importance. Domains have functional importance. Motifs perform similar functions to each other. Domains have unique functions. And finally, motifs are not stable independently. Meanwhile, domains are stable independently. So which of the following is correct about a motif? It is tertiary structure. Incorrect. This is correct for a domain. It's formed by disulfide bonds between amino acid chains. Incorrect. This is correct for a domain again. It's independently stable. Incorrect again. This is correct for a domain. It's made of greater than 250 amino acids. Now, I did not mention this directly, but try to take a guess. Tertiary structure, disulfide bonds, ionic bonds, stable independently. From this, you can kind of get the vibe that domains are larger in size compared to motifs. So, a new fact which we learned is that motifs are less than 250 amino acids, meanwhile domains are more than 250 amino acids. And then finally, it primarily exhibits structural stability functions, and this is the correct option. Question 5. Which of the following hormone receptors forms a heterodimer upon ligand binding? This is where it gets interesting. 3, 2, 1. Okay, let's explain. So we have two major classes of steroid receptors. The first one is steroid membrane receptors, which we're not interested about. The second one is steroid intracellular receptors. Now let's divide this class into two. Type 1 of steroid intracellular receptors are found in the cytoplasm. Okay. Everything which is highlighted could be a question. Okay. It's found in the cytoplasm. It's combined with HSP, heat shock proteins. Type 1 also forms homodimers. From the name, homo is same. So it forms dimers with receptors of the same kind. So examples are sex steroids and corticoids. So corticoid receptors are going to form dimers with corticoids exactly. It's not going to form a dimer with a sex steroid. And same for sex steroids, okay? Now, type 2 is kind of the opposite. It's found in the nucleus, that's the first thing. And it's often bound to DNA and co-repressors. Type 2 form heterodimers mainly with RXR. Even though they do form homodimers, the question is going to be which one forms heterodimers, if there's going to be a question, okay? So remember, homodimers type 1, heterodimers type 2. And the heterodimer is specifically with an RXR receptor. Homo is same, hetero is different, okay? So it's going to be either, let's look at the examples. Okay, the examples over here are thyroxine, vitamin A, and vitamin D. If we have a vitamin A receptor, it's always going to bind with an RXR receptor. If we have a thyroxine receptor, It's always going to bind with a RXR receptor. It doesn't matter what it is, it's going to bind to a RXR receptor. Okay? Now let's solve the question. Which of the following hormone receptors forms a heterodimer upon ligand binding? Is it testosterone receptor? No, it's not, because testosterone receptors form homodimers. They're type 1. Thyroxine receptors is the correct option. They're type 2 receptors. And then estrogen receptors, type 1. Progesterone receptors, type 1. Glucocorticoid receptors, type 1. Let's move on to question 6. Which of the following receptors is associated with the longest hours to days signal response time? I'll show the answer in 3, 2, 1. So pretty easy, you just have to memorize it. Moving from slowest to fastest, we're first going to find the steroid receptors. An example is nucleus-initiated steroid signaling. You also have membrane-initiated steroid signaling, but it's not important. So steroid receptors, slowest, hours to days. And then you have GPCRs, TKRs, non-TKRs, and JAK-STAT receptors. They're all seconds to minutes. And the fastest ones are the ion channel receptors, and their signal response time is milliseconds. So if we were to solve this, the longest is going to be the steroid receptor. The shortest is going to be the ion channel receptor. And in between, we're going to have B and C. We didn't mention anything about cytokine receptors. So the answer here is D. Question 7. Which of the following is correct during aldosterone signaling? You guys should know the answer for this because we explained it already. So aldosterone binds to an aldosterone receptor on the plasma membrane. Incorrect. Aldosterone is type 1, so it's going to bind in the cytoplasm. Okay, this kind of gave the answer. Aldosterone binds to an aldosterone receptor in the cytoplasm, so option B is correct. And then aldosterone binds to an aldosterone receptor inside the nucleus. Incorrect. It would be correct if aldosterone was a type 2 receptor. However, aldosterone is type 1. Okay. Aldosterone receptors form heterodimers with RXR receptors. Incorrect. That's for type 2. And then lastly, aldosterone receptor binding to heat shock proteins promotes translocation to the nucleus. Now, even though aldosterone receptors are bound to heat shock proteins, as we said before, this doesn't happen. The promotion of translocation to the nucleus is incorrect. Heat shock proteins actually prevent that. So when we get an aldosterone molecule, it binds to the aldosterone receptor, it moves the heat shock protein away, and when the heat shock protein the heat shock protein leaves, then there's translocation to the nucleus. So it's quite the opposite, and that's why E is wrong. The answer here is B. Now question 8. Which of the following is correct during vitamin D signaling? 3, 2, 1. Okay, first of all, we have to understand vitamin D receptors are type 2 receptors. So vitamin D binds to a vitamin D receptor on the plasma membrane. Incorrect. In the cytoplasm, incorrect. And then inside the nucleus, correct. So the correct option is C. Now let's look at those, D and E. Vitamin D receptors form heterodimers. We're almost correct. We're correct until now. And then with RAR receptors, it's incorrect. We said always with RXR. And then option E says vitamin D receptors bind to heat shock proteins in the cytoplasm to promote ligand receptor translocation to the nucleus. Incorrect. We don't bind to heat shock proteins, anything. It does bind to co-repressors though. This is it for cell signaling 1, 2, and 3. Take a quick break and be refreshed for molecular techniques. Okay, we're back. So let's begin with molecular techniques 1, lecture 4. And honestly, very easy. Okay, before we begin, I just wanted to say this is a very, very, very compressed review on molecular techniques. If you want the full package, I recommend watching my previous PAL, which is the entire molecular techniques thing in 1 hour and 10 minutes. Should be good, should be inclusive, inshallah. And you should focus on post-midterm more than pre-midterm. So don't go and start studying the slides from the beginning, all of that. I don't think you have time for that. Now let's actually begin. Again, DNA analysis, you have three core pillars: restriction endonucleases, cloning of DNA, and probes. What are restriction endonucleases? They are enzymes which cleave DNA into smaller, more manageable fragments, and they're used for paternity testing. Something about them is that they're very specific to restriction sites, and most of those restriction sites are palindromes. Now, cloning of DNA, this is basically where we amplify DNA by inserting this DNA fragment into a plasmid, and then we insert this plasmid back into a bacterial cell. The bacterial cell is gonna replicate, and the plasmid is gonna duplicate with the bacteria. And since we duplicate the plasmid, we're technically duplicating the DNA itself. And then we just collect those plasmids and extract the DNA from it. Okay. And finally, we have probes. Probes are pretty easy. We explained them a lot. And they basically bind to DNA, RNA, or proteins, and they give off a signal. This is the highlight of a probe. It gives off a signal. And we have different types of probes: double-stranded DNA, single-stranded DNA, and RNA and antibodies. They're all probes, or can be probes. Now, this is important, and I think you're going to get a question on this, hence the star. Fish, the fish technique. It's a technique which detects the number of copies of a gene in a cell using cDNA probes. What I mean by this is that if you have amplification of genes or deletion of genes, this is going to be detected using the fish technique. Again, we mentioned previously that fish is used to detect for HER2 positive breast cancer. And over here we have a breast cell, okay? We don't know if it's cancerous or not. We use the fish technique and we visualize the number of um, genes present over here, okay? And it specifically detects the epidermal growth factor 2. Normally, you're supposed to have two reds and two greens. If you have more than that, there's gene amplification. If you have less than that, you have gene deletion. And with HER2 positive breast cancer, the breast cancer cells tend to amplify the HER2 gene, and therefore we can see it a lot over here. We have seven red dots, which means we have seven genes, HER2 genes present, okay? All you need to know is in a normal cell, you have two reds and two greens. More than that is gene amplification. Less than that is gene deletion. And with HER2 positive breast cancer cells, there's gene amplification. Now, this is low yield, but I would still recommend if you review it. Dot blots, they use allele-specific oligonucleotides. And gel electrophoresis, you got questioned on this so much in the midterm. I don't think you're going to get questioned in the final. But even then, if you have time, go and review it. Now, for molecular techniques question, I think we have one question only. When using FISH technique to evaluate a breast cancer sample, which finding, what finding in a cell would indicate that it is a normal cell rather than a HER2 positive breast cancer cell? Okay, 3, 2, 1. The answer is gonna be C. Because in a normal cell, you're going to find two red signals and two green signals. So A, the presence of a gene deletion with only no, the amplification of the HER2 gene shown by 7, no, that's for a HER2 positive, not a normal cell. And then the use of a cDNA probe to detect an altered number of gene copies. There is no altered number of gene copies because it's a normal cell. Now for molecular techniques 2. I think this is the only slide we have for molecular techniques 2. It's so high yield. You're definitely going to get a question on this. So transfer analysis techniques. Just memorize this mnemonic and copy these and paste it into here. You're not going to get anything out of these. So we use southern blots for DNA, and some examples of that is RFLPs and VNTRs. They're all related with DNAs. RFLPs is the restriction fragment length polymorphisms. We're going to look into it, I think. And VNTRs are the repeats. They're all DNA. They're all related to DNA, And if you see them linked to any of the blots, it's going to be southern blot. Now we have northern blot after that, and it's RNA. Western blots used for proteins. So again, southern blots used for DNA, northern blots used for RNA, western blots used for proteins. Snow drop. Write the words on top of snow on top of drop, and then match it. S with D means southern with DNA. N with R, northern with RNA. Cancel the O's. W with P, western with protein. Now, let's test your knowledge on this. They're not going to tell you DNA. They're going to be sneaky about it. They're going to say something about, for example, if they say a transcript, first of all, you're going to think of having the DNA, then transcription happens, you then have mRNA. So it's going to be an RNA. The answer should be northern blot. What if they say after translation? After translation, you have a protein. So the answer is going to be western blot. And what if they say an enzyme? The answer is going to be western blot again, because enzymes are proteins. Now let's solve a question on this. Which of the following laboratory techniques is correctly matched with the macromolecule it's designed to detect and analyze? 3, 2, 1. The answer is, okay, let's go through them. A, northern blot with DNA, incorrect. Northern blot is with RNA. Western blot with RNA, incorrect. Western blot is with proteins. Southern blot with protein, incorrect. Southern blot is with DNA. And northern blot with RNA is the correct option. Now for molecular techniques 3, applications of VNTRs. Okay, so we're not going to talk about RFLPs, we're going to talk about VNTRs. Anyways, VNTRs stands for variable number tandem repeats, and I think they're going to question you on the clinical aspect of it this time. So let's look at what happens actually. Normally, you'd get a copy of the gene, you'd get a chromosome from your mother, a chromosome from your father, and therefore you're going to have two different copies of genes. And therefore, you're going to have two different numbers of VNTRs. One which matches your mother, one which matches your father. And if you sequence your genes, you're gonna find that if your parents are actually your parents, you're gonna find that the mother matches partially with the child, and the father matches partially with the child. This is correct matching, okay? Now let's look at this one. The mother matches the child matches with the father. That's correct. If they don't, then something is wrong. And they're also used for DNA fingerprinting because we all have a unique number of VNTRs and a unique sequence of VNTRs. They can basically take some DNA from the crime scene and then they can match it, the VNTRs, with the suspects, and we can clearly see that suspect 2 matches the crime scene DNA. And this is an application of VNTRs. Now let's talk about PCR. PCR stands for polymerase chain reaction. It's a series of reactions that amplify a specific DNA sequence. And the first reaction is denaturation, which is at a very high temperature, followed by annealing, which is at a relatively lower temperature, and primers anneal in the step. And then we have extending, which is in a medium temperature, 70 to 75. It's still high, but relatively it's medium. And Taq polymerase extends primers in this step. That's why it's known as extending. And Taq polymerase is a heat-resistant DNA polymerase. We will meet here again. So, okay. There are a couple of things which are needed for PCR to actually start. The first thing is the DNA which you're trying to copy. The second one is primers. And don't worry about the details. I don't think they're going to ask you. But read them on your own. The second one is primers. And then we have Taq polymerase, which is the heat-resistant DNA polymerase. And finally, we have the DNTP mix, which is the building blocks, the A, the G, the C, the T. Okay. Now, we can see over here that with PCR, the DNA strands increase exponentially every cycle. So if you start with 2, you're going to have 4 at the end of second cycle, 4 at the end of second cycle, and then 8, 16, 32, okay? That's an idea you should just get from PCR, okay? And then there's a couple of types of PCR. The first one is conventional or endpoint PCR, and it's qualitative. It tells you if DNA is present or DNA is not present. An example of this is, we're going to go through it in a bit. This is conventional PCR. It either tells you if the DNA is present or if it's not present. It doesn't give you a value. And then real-time PCR is different. It stands for real-time PCR, also known as qPCR, okay? Quantitative PCR. And quantitative, it tells you the number of copies. It gives you a relative or the actual number of copies. And this is the more advanced type of PCR, and it has probes that generates fluorescence. Um, these are some precautions for PCR. Just read through them. And some important keywords. The way I'm thinking of your final, it's going to be more clinical, more application based. And so they might actually question you on this. Some keywords, if you find them, the answer is immediately PCR. Prenatal diagnosis, preimplantation, genetic diagnosis, IVF, chorionic villus. And a note, RT-PCR stands for reverse transcription PCR. This has to go. Let's wait for it. Okay. RT-PCR is reverse transcription PCR and does not stand for real-time PCR. RT-PCR, you use RNA and you convert it back to DNA. Okay. Now let's look at this example of conventional PCR. Um, Reagent blank, you're not supposed to see anything. And then this is a sample which is known to be negative for EBV. EBV is Epstein-Barr virus. We're testing specimens for Epstein-Barr virus, okay? And then this is a sample known to be positive. So until now, we know that the test is accurate. And then we have two samples from specimen 1. They both test positive for ABV. Two samples from specimen 2, they both test positive for ABV. So from this, we can say that specimen 1 has EBV, okay? But it doesn't end here. We do another test with a protein known as beta-actin. And beta-actin is supposed to be found in every single cell of the body. So if the sample does not show beta-actin, it means we don't have a proper sample, okay? Now let's look at the results. With a negative sample, of course, you're not going to see beta-actin. Positive sample, you're going to see beta-actin. Specimen 1 and specimen 2. Specimen 1 showed tiny, tiny, tiny bit of beta-actin, and specimen 2 did not show beta-actin at all. So this clearly shows us that the sample has not been collected properly, and these results are not accurate. Okay? Okay, let's move to real-time PCR or qPCR. And they have two different probes which you should know about. The first one is Cyber Green. Excuse me. The first one is Cyber Green, which is DNA based. And then we have Tachman, which is cleavage based. And what this means is very simple, actually. Over here, we can see that the DNA is forming. This is the annealing step, denaturation, I mean. And the DNA are basically coming closer to each other, annealing. And then the moment the primer binds, you can see that the cyber green dye or the primer or the probe, I'm sorry, binds to the double-stranded DNA. And then it gives us a light signal. So we can basically detect the signal and know the amount of double-stranded DNA which is being formed. And from this, we can predict the number of double-stranded DNAs we have, okay? So we're gonna have, in the annealing step, when the primers bind, we're gonna have some little signal because technically it's still double-stranded DNA. And then in the extension step, when we actually have double-stranded DNA forming, we're gonna have way more cyber green dye giving us fluorescence, and we can detect that. Now for Tachman, which is cleavage based, the Tachman probe is going to bind to the single-stranded DNA in the beginning. And then as the DNA strand is growing, it's going to reach this probe, it's going to cleave it. And at the two different ends of the probe, you have R and Q. R is a reporter and Q is a quencher. What R is, it basically gives off a light signal, okay? So R is shining. And then Q is absorbing all of that. So we cannot detect it. But the moment they're separated, Q goes away and R goes away, R can actually, it's like they're giving us a lesson. R can actually shine. It can give off the fluorescence, and we can detect this fluorescence, okay? So if the probe has been broken, it means double-stranded DNA has been formed, and we can detect this fluorescence from R, okay? Okay, now let's talk about real-time PCR analysis, and the graph may look confusing. It's pretty simple though. The first phase, this horizontal line, is the lag phase, okay? And even though we have uh, increasing copies, the machine cannot detect it. The fluorescence is too low for the machine to detect. Now, after that, we reach the log phase, which, as you can see, the graph is going up. And over here, we have exponential growth, and the machine detects it. And then after that, we have the plateau, and the reason is unknown. Don't worry about it. And one more thing I want to mention over here is the CT, or threshold cycle. And threshold cycle simply means the number of cycles needed to reach the threshold. And memorize it if you don't understand it, but the higher the number of DNA copies we start with, the lower the number of cycles we need, and vice versa. Okay, let's move on to few low yield concepts. They're low yield, I'm not going to mention them, but if you have time, review them. The first one is Sanger sequencing. You got questioned a lot in the midterm. I doubt you're going to get some questions in the final. Then microarrays, they're not part of your curriculum anymore. They shouldn't be reviewed actually. Incorrect heading. But I wanted to address this confusion because a lot of people asked me about my slides last time. The slides were prepared before your last lecture, and so I had to include microarrays just to be safe. But the doctor said you're not required to know microarrays, so don't study microarrays at all. And then COVID-19 tests, they're low yield, but if you have time, review them. Now, question one. Couple undergoing IVF opts for pre-implantation genetic diagnosis to screen embryos for a specific, just you have to know it already, okay, without reading the question. For a specific single gene disorder before uterine implantation, which of the following laboratory techniques is most commonly utilized to rapidly amplify and detect the targeted gene sequence from a single biopsied embryonic cell? The answer is B, PCR. Don't, you just have to memorize this, okay? And we're done with 6 lectures, 6 more to go. Um, we're at the halfway mark. I recommend you take a break and like 5 minute break. See you after that. Okay, let's continue. So, how do enzymes work? Enzymes are basically proteins which reduce the free energy of activation needed for action to take place. And this is the free energy of activation. So the purple line is with the enzyme, and the blue line is without the enzyme. If you're colorblind, this line is without enzyme, with the enzyme, I'm sorry, and this line is without the enzyme. And as we can see, the only difference the enzyme does is it reduces this energy needed for the reactants to reach this line or this line. And this line is, or this state, the peak state, is known as a transition state. Okay? So, quick recap, enzymes are used to reduce the free energy of activation needed for a reaction. And the free energy of activation is the energy which takes the reactants from their starting energy to the peak, which is the transition state. Okay? And after that, they're all gonna undergo the reaction with an enzyme or without an enzyme, and they're gonna become products. The difference in energies between the reactants and the products is known as the free energy of the overall reaction. And very important point to remember is that if you use an enzyme or if you don't use an enzyme, if you actually end up moving from the reactants to the products, the free energy of overreaction remains unchanged. There's no change to it. Simply, it's products energy minus reactants energy. No change. So there's no effect on the over. I'm just waiting for this to move. Read the bottom line. There is no effect on overall reaction energy change. It's also known as Gibbs free energy. So Gibbs free energy remains unchanged. Now, there are six major classes of enzymes, and they have different reactions which they catalyze. The first one is oxidoreductases, and from the name, you can think of oxidation and reduction reactions, or redox reactions if you know them. And we can see that A plus NAD plus gives us a B plus NADH. Just remember, oxidation reduction is oxidoreductases. Now, reaction two is transferases, and from the name, transfer. So you're gonna have A and B. You're gonna transfer one part to the other and one part to the other. This is gonna give us two new compounds, which are C and D. So A plus B gives us C plus D. And then we have ligase and lyase. Ligase comes from the word ligate, which is joining together. So we're going to have A and B, join them together. They're going to give us C. And lyase is the opposite. You're going to start with A, for example. You're going to break it apart, giving us B and C. And then we have hydrolases. From the name, hydro is water. And then the remaining part, just remember water, and water is used to cut something, okay? Hydrolase. So water is used to cut compound A. It's going to give us compound B and compound C. So A plus H2O gives us B plus C. And finally, isomerase. I'll use this Enderman plushie to help me explain. So isomers are basically they have the same number of atoms and same number of bonds and okay, not necessarily same number of bonds, same number of atoms, but they're just arranged differently. So if we start with A, you're gonna end up with a different A, like a flipped A or whatever, it's gonna be B. So if we have this Enderman plushie over here and we take its head off and attach it to its side, it's still gonna be like the same number of atoms totally, but it's gonna be a different look, and therefore they're gonna be isomers. So we're going to start with one compound, we're going to end with one compound. However, they're different, no new atoms added to the entire thing. Now, this is important, and you may get a question on this, on any of those. Remember, quick review, oxidation reduction, oxidoreductase, transfer, transferases, cut, join, ligase, cut, lyase, cut with water is hydrolase, and change the looks or shape as isomerase. And then we have zymogens, and zymogens are the inactive precursor of an enzyme. And they usually end with the suffix gen. So an example is pepsinogen, which gives us pepsin. Now let's look at what affects enzyme activity. We have two important factors, ignore the third one. Their temperature and pH. And one thing about pH, before that, let's talk about how they affect enzyme activity. If you have a very high temperature, enzymes will denature and they're not going to work anymore. If you have very extreme pH, either very low or very high, enzymes will also denature. If you have a very low temperature, the enzyme will be inactive, but it will not denature. Now let's talk about Q10, which is the temperature coefficient, and it simply stands for the multiple by which the reaction rate increases when we increase the temperature by 10 degrees. So let's look over here. At 30, the enzyme activity or the reaction rate was over here. If we increase the temperature by 10 degrees and take this recording, we can see that it's 8. So the first one was 4, this one was 8, it doubled, and so the Q10 or the temperature coefficient is 2 because it's the multiple. Okay? And then let's look at this enzyme activity over pH graph. And we can see two different enzymes. They have two different optimum pH values, and they denature when the pH values are extremely away from their optimum values. Okay? Now, this is an overcomplicated concept for some reason. I don't know why. Very simple, just focus with me. Firstly, we have a part of an enzyme, the protein portion, okay? And it's inactive, of course, because it's only a part. And this is known as an apoenzyme. Okay, so protein portion of the enzyme inactive is the apoenzyme. Then we have the coenzyme or the cofactor, and it's the non-protein portion of the enzyme, also inactive. And then they come together and they form this holoenzyme, which is protein plus non-protein portions. It's the whole enzyme and it's active. Okay? Very simple. You start with the protein part, apoenzyme. The non-protein part, the cofactor or the coenzyme, it binds, and then now you have the holoenzyme. And then after that, you can add the substrate and whatever. Now let's look at different types of holoenzymes. The first one is apoenzyme with a coenzyme, and an example of coenzymes are specifically organic molecules, okay? An example of a coenzyme which is an organic molecule is NAD+. And then we have prosthetic groups, which are tightly bound coenzymes. So you're going to have the apoenzyme bound tightly with a prosthetic group, and the prosthetic groups are coenzymes, however, they're tightly bound. And an example of this is FAD+. And then we have apoenzymes bound to metal coenzymes, and the metal coenzymes are known as cofactors, okay? Three different types of holoenzymes, pretty simple. Now let's talk about isoenzymes. In one line, they're the same. They have the same enzyme activity, but they have different amino acids sequences, and they're coded by different genes. Remember this: same enzyme activity, but different amino acid sequence, coded by two different genes. Now, two graphs I want you to remember the shape of. Don't understand, just remember it. The substrate concentration over the reaction velocity. When you don't have an enzyme and the reaction is taking place, this is going to be the shape of the curve. It's going to be linear. But when you have an enzyme, it's going to be a hyperbolic shape, okay? You're going to have this exponential increase, and then slowly it's going to plateau where it reaches the Vmax. Now, question one. Specific biochemical reaction involves the rearrangement of atoms within a single substrate molecule, converting it into a structural isomer without adding or removing any atoms. Which class of enzymes is responsible for catalyzing this process? I'll give you 3 seconds. 3, 2, 1. Is it A, isomerase? Yes, it is. As we explained using this guy. This is exactly what isomerases do. Question 2. What is the effect on the overall reaction energy or Gibbs free energy after enzymes are used? 3, 2, 1. The answer is no change. As we said, Gibbs free energy does not change after enzymes are used, before enzymes are used, without enzymes. It always doesn't change. Now let's move on to lecture 8, Enzyme Kinetics. We have two important hallmarks, which are Vmax and Km. Vmax is the maximum velocity. And then we also have V0, which is the initial velocity. It's going to be over here. And then Km is the substrate concentration, as we can see, substrate graph, substrate concentration at half of the Vmax. So you're going to get the Vmax divided in half. And then if you find the substrate concentration, it's going to be Km. Or vice versa, okay? And Km is important because it's an inverse measure of affinity. If you think about it, if the Km, if you need more substrate concentration, or the Km was at a higher substrate concentration to reach the same Vmax, it means the enzyme and the substrate don't like each other that much. And so the higher the Km, the lower the affinity. And the lower the Km, the higher the affinity. Just memorize this, okay? Now, using this Km equals substrate concentration at half Vmax, they're gonna create this equation, okay? You don't have to create it on your own, you just have to memorize this format. And the reason they do this is because they want to include everything in a straight line, in a linear graph. Because when it's a linear graph, you can take the y-intercept and the x-intercept, and they're gonna give you the Km values if you try hard enough, okay? So from this, we can see that if you read it, the intercept, the y-intercept, the place where the graph crosses the y-axis, is equal to 1 over Vmax. So whatever value you get over here, let's say you got 2, you're going to do 1 divided by 2, which is going to give you 0.5, and this is going to be the Vmax value. Let's say you got this value, and this value it's to the left of the y-axis, so it's going to be negative, okay? So 0, 1, 2, 3 to the right, and then 0, minus 1, minus 2, minus 3 to the left. So let's say you got minus 3 over here. You're first going to multiply it by minus 1, get rid of the negative. And then you're going to do 1 divided by this value, which you got over here. This is going to give us 1 over 3, and therefore the Km is 1 over 3. You just have to memorize it, honestly, I can't do much. And then there's a couple different types of feedback, which you should know. The first one is negative feedback or positive feedback. And this is basically when a product of a reaction comes back and tries to slow down the reaction. This is going to be negative feedback, okay? Positive feedback would be if it goes back and it makes a reaction faster, okay? Now, if we take this negative or positive feedback and we couple it to another reaction, it's gonna give us a different type of feedback. So let's look at this. A is converted to B, B is converted to C, C to D, D to E. And then when E is converted to CO2 and H2O, ADP is also converted to ATP. Now, this ADP is gonna go and give a positive feedback to this enzyme. This ATP is going to go and give a negative feedback to this enzyme. Okay? And then finally, we have forward feedback, in which the substrate itself influences the enzyme to increase its rate of reaction or whatever. Okay? It's pretty low yield, this one. Now, let's talk about allosteric enzymes. Allosteric enzymes are enzymes that are regulated by the products of the pathway they control. Basically, they control this pathway. They convert A to B. One of the products in this pathway is gonna control it. Remember this, okay? One of the products in this pathway is gonna control it. So it thinks it's in control, but it's technically being controlled, okay? And one more thing about allosteric enzymes is they also catalyze the committed step. You should remember this from MOL 1, committed step, rate limiting step, same thing. And they have different curves when we plot the substrate concentration and the reaction velocity. The green one is the allosteric enzyme, and it shows a sigmoid curve. The blue one is the Michaelis-Menten enzymes, and they're not controlled by products of the reaction, and they show the hyperbolic curve, which we saw before. Now we have a couple of enzyme inhibitors, irreversible class, and then reversible. The reversible class is subdivided into 3. Competitive, where the inhibitors bind to the active site. Non-competitive, where the inhibitors bind to the enzyme but not the active site, or they bind to the substrate. And then you have uncompetitive, where the inhibitors wait for the substrate to bind to the enzyme, and then they hold it all together. So this is the enzyme, and then substrate comes in, and then the inhibitor is going to come and hold all of them together. So the substrate cannot leave. And this is uncompetitive inhibition. And then these are the different graphs of them. You should memorize all of them and know why they look different, okay? So for competitive inhibition, we can see that the x-intercept is closer to the 0. And so the closer it is, the higher the Km and the lower the affinity. Because if it's closer, it means the value of 1 over Km is going to be less. And so the value of Km is going to be more. If I'm not making any sense, I have a summary table for you to memorize, okay? But it's pretty simple. And the Vmax, the Vmax is not changing at all because the y-intercept is the same. And then over here, the Km is not changing because the x-intercept is the same, but the Vmax is changing. The y-intercept is higher in this example, and so the Vmax is going to be lower. It's always the opposite. Km is the x-intercept is smaller, Km is going to be higher, the y-intercept is higher, Vmax is going to be smaller. And then with uncompetitive inhibition, you have a smaller Vmax and a smaller Km. And the only one which, the only type of inhibition from those which is improved with increasing the substrate concentration is the competitive inhibition. This is a summary table. Let's go through it quickly. With competitive inhibition, the binding is to the active site. With non-competitive inhibition, the binding is to the enzyme or the substrate. And with uncompetitive, the binding is to the enzyme substrate complex. With competitive inhibition, the Vmax is unchanged, the Km increases. Non-competitive, the Vmax decreases and the Km is unchanged. And with uncompetitive, the Vmax decreases and the Km decreases. Both of them decrease. And then with competitive inhibition only, it can be overcome by increasing the substrate concentration. Now, let's solve this question. Which type of inhibition is shown in this graph? 3, 2, 1. Okay, let's interpret the graph first. We can see that the Km did not change, but the Vmax decreased because the y-intercept is higher now. So the answer is B, non-competitive inhibition. Now for the last enzymology lecture, clinical enzymology. We're almost done, guys. So we have 3 different clinical applications of enzymes. The first one is diagnostic, second one is therapeutic, and laboratory. Don't worry about therapeutic. For laboratory, just remember ELISA. And then for diagnostic, we're going to talk a lot about this. The entire lecture is basically about diagnostic applications of enzymes. And they focus on plasma enzymes. So they measure the plasma enzymes levels. If they're less than normal, then this is an inborn error of metabolism. It just means you cannot produce this enzyme. If the level of the plasma enzyme is more than normal, it could either mean there is cell damage, so the cells are damaged and they're leaking these enzymes into the blood, or there's cell proliferation. And we're going to focus on different examples of cell damage. The first enzyme the enzyme class which we're gonna talk about today is creatine kinase. And we have 3 isoenzymes of creatine kinase. First one is CK1 or CKBB, and it's elevated in brain or CNS diseases. Water break. Okay, so CK1 or CKBB is elevated in brain or CNS diseases. CK2 or CKMB is elevated in acute MI or myocardial infarctions, so heart, okay? Then CK3 or CKMM is elevated in muscle damage. All of these are important. You have to link it together, okay? So if I ask you which one is elevated in heart diseases or in acute MI, it's going to be CK2 or CKMB. You should also know both names. Now, let's talk about the LDH isoenzymes. We spoke about the CK isoenzymes. Let's talk about the LDH isoenzymes. Now, the total LDH value is non-specific because each LDH isoenzyme points towards a specific marker, okay? So LDH1 is cardiac specific, so we're going to see a rise in MIs, for example. LDH2, LDH3, don't worry about them. LDH4 and LDH5 are liver specific. So you're going to see an increase in a liver disease, for example. Now, application for you guys. If this is normal, normal LDH1, normal LDH2, normal LDH3, 4, 5, what would this be? Think about it. So let's compare the LDH5, 4, 3, 5, 4, 3. They appear to be normal, relatively. LDH2 to LDH3, relatively the same. LDH1 is more than LDH2, clearly indicating LDH1 is increased. And so LDH1, cardiac specific, MI. These are how the questions are going to be. Now let's talk about cardiac markers. Very, very, very high yield, very high yield. For cardiac markers, you have three main cardiac markers which are used nowadays: myoglobin, troponin I, and CKMB. CKMB is also CK2. Now, troponin I is the most specific cardiac marker. This is a question, guaranteed. Most specific cardiac marker, troponin I. And let's talk about the values over here because they're gonna like point us in the direction of when to use myoglobin, when to use troponin I, when to use CKMB. Myoglobin is used in early diagnosis because it's the first cardiac marker to increase. Troponin I is used for late diagnosis because it goes back to normal values after the longest time. And just remember CK2 or CKMB for recurrent MIs. Okay? Now, as for the values, I hate to say it, but you have to memorize them. You could get a question on this. What happens um, if we're like 3 hours? 3 hours is going to be myoglobin. If it's more than 3 hours, then you could use any of them. If it's more than 4 hours, you could use any of them. And then if they mention anything about a few days later, 6 days or whatever, 4 days, for example, it's going to be troponin I because it's used for late diagnosis. So you have to memorize the values. Quick recap: myoglobin, early diagnosis; troponin I, late diagnosis; CK2 or CKMB, recurrent MI. Question time. Which biomarker is expected to be elevated in an individual who experienced a recurrent heart attack within 6 days? Okay, so keyword recurrent. The answer is going to be CKMB. Okay, again, CKMB, recurrent MI. Troponin I, late diagnosis; myoglobin, early diagnosis. What is the main reason that troponin is preferred over creatine kinase in the diagnosis of MI? 3, 2, 1. This one, you just kind of have to remember it. It gets repeated a lot as well. The answer is E. Troponin is more specific than creatine kinase. As we said before, troponin I is the most specific cardiac marker. And lastly, we have this very, very important enzyme table, and I believe 2, 3 questions from this. So the first enzyme is acid phosphatase, and the source is the prostate only. So the only diseases in which you're going to see a rise in acid phosphatase is going to be prostate related, which are prostate cancer and prostatitis, inflammation of the prostate. I think this is going to be a question because it's newly added to your slides. And then you have alpha amylase, which is produced by the pancreas and the salivary glands, and therefore the diseases are going to be acute pancreatitis and mumps. Then you have lipase, which is only produced by the pancreas, and it's going to be increased in acute pancreatitis. Now, let's say they give you a question says you have an increased value of alpha amylase and you want to determine if it's a pancreatic issue or a salivary gland issue. What other test result could you order? The answer is going to be lipase because if you get the lipase test results and it's not increased, it means it's not a pancreatic issue, it's a salivary gland. However, if lipase is increased, it's going to be a pancreatic issue. Okay. Now let's move on to gamma glutamyl transpeptidase GGT, and the source is the liver, and therefore it's elevated in liver diseases. And then we have ALP. ALP, the source is the liver and the bone, and therefore it's elevated in liver and bone diseases. Just like we explained this alpha amylase and lipase example, a question could come in GGT and ALP. And then we have ALT and AST. You guys know them a lot by now. It came in the midterm, I think. And ALT is produced in the liver, AST is produced in the liver and the heart. And the important thing you should know for them is that if the ALT value is increased more than the AST value, this is indication of viral hepatitis. However, if AST is more than ALT, It's alcoholic liver disease. The way I remember it is AL. The L reminds me of alcoholic, but it's quite the opposite. So if AST is more than ALT, it's alcoholic. Then the other one is viral hepatitis. I'm sorry if I confused you with this one. Question three. A woman presents to your office with elevated ALP levels and bone issues. Which additional test should you consider? So remember the options: elevated ALP levels and bone issues. ALP measures for liver and bone. So you want a test which is going to tell us, is it a liver only or a bone only? Let's go back. Do we have any other bone only? No, we don't. So we need to find a liver only test. It's either ALT or GGT. And the answer is C, GGT. Okay? Pretty straightforward, but you have to remember each individual enzyme and the sources. And with that, we're done with the enzymology lectures. One last push. I know you guys are burnt out already, but let's finish the last three lectures. They're very, very simple, and we can finish in a few minutes, inshallah. So insulin is, as you guys should know by now, insulin is the anabolic hormone, and it's the hormone which is increased in the fat state. So by anabolic, I simply mean it builds. They say anabolic steroids, whatever. It builds muscle, okay? So insulin is gonna always be building. It's gonna build, 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 build. And the source of insulin is the beta cells in the pancreas. The trigger for insulin's release is increase in blood glucose, increase in amino acids, and increase in incretins such as GLP-1 and GIP. And the effects, as we said, anabolic effects, which are building or storing. And then something which is unique about insulin is that it's produced in equimolar amounts with something known as C-peptide. The C-peptide is a part of the insulin precursor. So you're gonna have proinsulin, and then, okay, let's say the structure is actually somewhere like this. So you have proinsulin, it's cut, insulin goes in the body, and C-peptide also goes in the body, okay? So they're produced in equal amounts, and the importance of C-peptide is because it has a longer half-life, and it's an indication of endogenous insulin. So I'll give you this scenario. Let's say we have a diabetic patient and we inject him with insulin. Okay? We want to measure the amount of insulin his body produces, not the amount of insulin he has as a whole. If we measure insulin, it's going to be inaccurate because we give him exogenous insulin as well. But remember, we said there's something which is produced in equal amounts with insulin and it has a longer half-life, which is the C-peptide. And so it's the best marker of endogenous insulin. Okay? Now let's talk about glucagon. Glucagon is the opposite of insulin. It's the catabolic or the fasting hormone. The source is the alpha cells of the pancreas. A trigger which results in the release of glucagon is decrease in blood glucose, increase in amino acids, and increase in stress. Now, if you can recognize this, an increase in amino acids is common in both. So when you get increased amino acids, or when you eat a protein-rich meal, you're going to increase both insulin and glucagon. For different reasons, of course, but you're going to still increase insulin and glucagon. And the effects of insulin is catabolic, which is breakdown or releasing. And we spoke about a unique thing related to insulin, which was C-peptide. The unique thing over here with glucagon is, okay, I'm not going to point down, just focus on the last one, which is stress. In times of stress or when the body is stressed out, you release epinephrine and norepinephrine, the catecholamines. And what they do is that they override insulin, even if the blood glucose is high, okay? Low, high, whatever, the body doesn't care. If you release epinephrine, this is going to override the function of insulin and you're going to stimulate glucagon. And then glucagon is going to release glucose, okay? So usually we release epinephrine, norepinephrine, when we're like in sports or when we're stressed out, when we need the energy basically. And that's why insulin function is overridden by the epinephrine, and we stimulate glucagon, and then we increase the blood glucose. And now this one says that they will both work against their triggers. Let's look at the triggers of insulin and see what's going to happen. So a trigger of insulin is increase in blood glucose. So insulin is going to try to decrease the blood glucose. Amino acids. Increase in amino acids is going to trigger insulin. So insulin is going to reduce the amino acids, and it does that by increasing protein synthesis. And incretins, don't worry about that. Now for glucagon, decrease in blood glucose, it's gonna try to increase the blood glucose by releasing glucose. Increase in amino acids is gonna decrease or try to aim to decrease amino acids. Um, for example, one example I could think of is using the amino acids in gluconeogenesis, for example. And stress, don't worry about it. Now, let's quickly review some of the anabolic and catabolic reactions. You don't really need to know the details. I just want you to remember them from MOL 1, okay? Because it's going to help you with the functions of insulin, functions of glucagon, metabolism in diabetes, all of that later, okay? So the first one is by the way, most of the anabolic reactions are going to be promoted by insulin, except gluconeogenesis. And most of the catabolic reactions are going to be promoted by glucagon. Is there any exceptions? There is no exceptions. So gluconeogenesis, if gluconeogenesis is put over here, there's going to be insulin reactions, glucagon reactions. Okay? So let's talk about each reaction individually. Glycogenesis. It means formation of glycogen. We are building glycogen, it's anabolic. Then you have protein synthesis. Again, anabolic because you're building. And then TAG synthesis, which is the triacylglycerol synthesis. You're building the three fatty acids and glycerol backbone thing. It's anabolic. Then gluconeogenesis, you're forming new glucose, and therefore it's anabolic. And then you have the catabolic reactions, starting with glycogenolysis. Break it down. Glycogen lysis means you're breaking down glycogen, catabolic. Ketogenesis. This is formation of ketone bodies. Now, even though it's formation, you form it from fatty acids, and fatty acids are larger. So you're going to get the fatty acids, break it down, and this is going to form ketones. So even though it's a formation reaction, kinda, it's still catabolic. It's an exception, just memorize it. Then you have fatty acid oxidation. You're breaking down fatty acids. This is catabolic. Then protein breakdown, catabolic. Lipolysis is also catabolic because you're breaking down lipids. Now, transporters, very important, very high yield. Don't worry about GLUT3 and GLUT5 though, because the remaining are very important and very, very high yield. The first one is GLUT1, and it's found in RBCs and in the blood-brain barrier. Okay? The second one is GLUT2, and it's a glucose sensor. It's found in the liver and in the pancreas, in the beta cells. So the beta cells are gonna have this GLUT2. They can sense the glucose level in the blood. When it increases a lot, they can release insulin. Okay? And then GLUT3 skip. GLUT4 is insulin dependent, and it's found in muscles and in adipose tissue. And the importance of this is that when insulin rises, it means basically it goes from GLUT2 to GLUT4. First of all, the sequence starts when we increase our blood glucose. This is detected by the pancreas through the GLUT2 sensor. And then it's going to release insulin. This insulin is going to go through the blood. It then goes to the muscle cells or the adipose tissue. And then it's going to recruit the GLUT4 to the cell membrane. And when it recruits the GLUT4 to the cell membrane, this is then going to allow glucose to enter the cell. And therefore, this is insulin dependent. It's not always there. It's only going to be there when there's insulin. And then we have GLUT5. Don't worry about GLUT5. So if a question asks you about which of the following tissues are insulin dependent, or they absorb glucose when insulin is present, and they give you liver, pancreas, um, RBCs, the brain, muscle. What's the answer going to be? The answer is going to be muscle because the others are not insulin dependent, they're insulin independent. The only one which is insulin dependent is the muscle from these options, also adipose tissue. Now let's compare insulin and glucagon. If I were to put stars like molecular techniques, I put 5 stars over here, 5 stars over here. Very important slide. Okay, so let's start with insulin. Insulin promotes glucose uptake. We spoke about this. It increases the GLUT4 expression, and GLUT4 transporters move to the cell membrane, and they allow glucose to enter the muscles and adipose tissue. Insulin also increases protein synthesis because it's an anabolic process. And as we said before, insulin and glucagon both act against their triggers. One of the triggers was increased amino acids in the blood. So when we have increased amino acids, we increase the protein synthesis. And then insulin also increases glycogen synthesis because, again, it's an anabolic hormone. So anabolic, simply think of it this way, that insulin and glucagon, insulin is when you're overfed. Insulin tells your body you have so much, let's store. And glucagon does the opposite. It tells your body you're starving, let's release some sugar into the blood. Okay, so where were we? We're over here. Okay, insulin increases glycogen synthesis, also known as glycogenesis. We spoke about it before, pretty straightforward. And then fat synthesis. Insulin increases fat synthesis, and it does this by increasing LPL. You should review this in your fat lectures. But LPL basically increases when we're building up fat. And then HSL increases when we're breaking down fat. So LPL increases with insulin, HSL decreases with insulin. LPL decreases with glucagon, HSL increases with glucagon. Okay, let's get back to insulin. Insulin increases fat synthesis because it's anabolic. And then insulin decreases ketogenesis. You just have to memorize this. You don't need to produce ketone bodies. And we're going to talk about this later in which lecture? Feed fast, okay? So ignore this for now. And then insulin decreases lipolysis. You don't need fats in the blood. You already have so much glucose. You instead want to build fats. You don't want to break down fats. And then you have gluconeogenesis, which is decreased when insulin is released. And it's pretty straightforward. You have so much glucose in the blood, you don't want to build new glucose. And glycogenolysis is also decreased. You already have so much glucose, you don't want to break down glycogen to give you even more glucose. And now glucagon is basically going to do the opposite of these functions. It's not going to affect glucose uptake because it's not going to affect the GLUT4 transporters. Glucagon is going to decrease protein synthesis because it wants to release these amino acids into the blood to the liver for the liver to use them in gluconeogenesis to produce new glucose. But it's not included here. It means it's not important for you guys. And then glucagon decreases glycogen synthesis. Again, it decreases glycogen synthesis because, or glycogenesis, because we don't have that much glucose in the first place. We're not storing now, we are releasing. And glucagon decreases fat synthesis because, again, we spoke about the LPL HSL. Remember this, insulin is going to increase LPL activity. Insulin is going to decrease HSL activity. Glucagon is going to decrease LPL activity. Glucagon is going to increase HSL activity. And LPL is used for fat synthesis, lipoprotein lipase. HSL is used for lipolysis. Okay. Okay, so glucagon decreases fat synthesis, and glucagon increases ketogenesis, and we will talk more about this in the feed fast cycle. Glucagon also increases lipolysis, and glucagon increases gluconeogenesis. The reason it increases gluconeogenesis is because, again, glucagon senses that we're starving. The cells need glucose. And so it's going to tell the liver, we need glucose no matter what, produce new glucose. Therefore, it's going to increase gluconeogenesis, formation of new glucose. And it's also going to increase glycogenolysis, which is producing glucose or breaking down glycogen to give us glucose, because again, we need the glucose. And finally, it's going to increase amino acid uptake. Just memorize this, okay? Now, what about protein breakdown? It's not included in this. Would insulin increase protein breakdown or not? The answer is no. Insulin increases protein synthesis. Glucagon would increase protein breakdown. And I kind of gave you the reason before. It's because we want the amino acid precursors to be used in gluconeogenesis. So we're going to break down proteins, get the amino acids, put them in the liver, and then produce new glucose. Okay, now let's move on to question 1. Which of the following best explains why C-peptide levels are measured clinically to assess endogenous insulin secretion? Three, two, one. There's not much explanation to do here. The answer is C. C-peptide is secreted in equimolar amounts with insulin, but has a longer half-life. That's why it's the best marker for endogenous insulin. Question 2. During periods of physiologic stress, such as fight or flight response, which of the following best describes the interplay between epinephrine and glucagon? 3, 2, 1. Okay, is it A, epinephrine suppresses glucagon? No, does the opposite actually. Epinephrine enhances insulin? No, does the opposite. Glucagon inhibits epinephrine release? No. Glucagon and epinephrine both promote insulin-mediated glucose uptake? No. Epinephrine stimulates glucagon levels, overriding the usual feedback from blood glucose levels. Correct. The answer is E. So the answer is C over here. The answer is E over here. Question 3. Which of the following is a correct effect of increased insulin levels? Increased gluconeogenesis in the liver, decreased glycogen synthesis in muscle, decreased hormone-sensitive lipase activity in the adipose tissue, increased lipolysis in adipose tissue, or decreased glucose uptake by cells. One of them is the effect of increased insulin. The others are the opposite. So let's go one by one. Increased gluconeogenesis in the liver. This is the opposite. The insulin decreases gluconeogenesis. Decreased glycogen synthesis in the muscle. Incorrect. We do the opposite actually. We want to store the glucose because we have so much of it in the blood. So we're going to store it in forms of glycogen. And then C, decreased HSL activity in adipose tissue. We said HSL breaks down, is involved in lipolysis. So this is correct because we don't want to break down fats. We already have enough fuel in the body. And then D is increased lipolysis in adipose tissue. Incorrect. It's decreased lipolysis. And then E is decreased glucose uptake by the cells. It's incorrect. We said the first point was increased glucose uptake. Now, lecture 11, feed-fast cycle. I say we just go straight into it. Okay, let's start. So for the feed-fast cycle, we have 3 main phases which you should know in details. The first one is the absorptive, oily fed state, and you should know both names, by the way, for each state. The absorptive or the fed state, and it's the first 3 hours after you eat the meal. So the hour 0 is going to be the moment you finished your meal, and then up to 3 hours after the meal. And in this state, you have a high insulin, low glucagon ratio. So an increased insulin to glucagon ratio, and you're in the storage mode. You're basically storing glucose, fats, whatever. And then in stage 2, the post-absorptive or the early fast stage, it's going to be 3 to 12 hours or maybe even up to 18 hours after your meal. And in this stage, insulin drops, glucagon rises. So the ratio of insulin to glucagon is gonna decrease a little. And the main aim of this is to maintain the blood glucose and to mobilize some glucose, to release some glucose into the blood. Okay? And then the last stage is the prolonged fasting or starvation, and it's more than 24 hours. And in this stage, you don't have any glycogen reserves. You finished your glycogen. Glucagon dominates. Insulin is going to be very, very low. Glucagon is going to be very, very high. And some key factors in this is fatty acids and ketones. They play a huge role in this stage. And you're just trying to adapt to whatever fuels you have remaining. So let's talk about phase one, the fat phase or the fat state. And as we said, it's the storage mode. So let's focus on what happens in each of those organs. The liver increases glycogenesis. That's the first thing. It also increases fatty acid synthesis using the excess glucose. So we're just storing the glucose basically. And it increases and wait, we said fatty acid synthesis using the excess glucose, and then it releases those fatty acids into the blood through the chylomicrons and VLDLs for the adipose tissue to do their meaning, okay? And then let's talk about the muscle. Muscles increase the glucose uptake. We spoke about this before. Muscles increase the glucose uptake, GLUT4. They increase glycogenesis and protein synthesis. So they're storing glycogen and they're forming proteins now. And one thing about muscles, which you should know, very important, is that they can store glycogen, but they cannot release glucose. And this is because they lack glucose 6 phosphatase, which sets glucose free. So even if they try to release the glucose into the blood when we're starving, they cannot do that. Okay? And then we have adipose tissue, and adipose tissue increases glucose uptake, again, GLUT4 transporter, and TAG storage. So they form triglycerides and they store them using the glucose, the excess glucose we have in the blood for glycerol, and the fatty acid from the chylomicrons and the VLDLs. That is it for adipose. And now for very, very, very key players in this entire feed fast cycle, the brain and the RBCs. Now the brain and the RBCs use glucose normally because this is what they can use. They can't use fatty acids, both of them. But we're going to see something interesting about the brain in the last phase. And if you remember, we said they are insulin independent. And what's their transporter? GLUT1. GLUT1 for the RBCs and for the blood-brain barrier. Now, question. What's happening to LPL activity and HSL activity in phase 1? 3, 2, 1. Okay. Remember we said phase 1 is insulin dominated. Insulin is high, glucagon is low. So the effects of insulin are going to take place. And we said insulin decreases HSL activity and it increases LPL activity because we don't want to break down fats. Instead, we want to store them. And therefore, this is the answer. Now let's move on to phase 2, the early fast. And this is when we start to خلاص, the glucose in the blood is basically running out and we're releasing glucose now. So in the first stage of phase 2, like the first part of phase 2, we said it's 3 to 12 hours, maybe even up to 18. Let's say we're like in the 3 to 6 hours. In that first stage, you're gonna have glycogenolysis because we still have glycogen reserves. And then after the glycogen reserves run out, we rely on gluconeogenesis. Again, one more time. In the beginning, we use glycogen reserves, break down the glycogen into glucose and release it into the blood. And then we rely on gluconeogenesis because we don't have any more glycogen. We have to form new glucose using fatty acids. Okay? Using the amino acid precursors we spoke about before. And in this stage, the liver survives using fatty acids. So the liver since it can use fatty acids, unlike the brain and the RBCs, it uses those fatty acids and it releases this glucose into the blood. Okay, it's basically being generous. And then for stage two, the muscles increase fatty acid oxidation. This is the first part. They increase fatty acid oxidation, so they're not using glucose anymore. They use fatty acids, and they release amino acids to support gluconeogenesis. So now, as you can see, all of the organs are thinking of the body, the body tissues, which cannot use anything else but glucose. And they're shifting their fuels from glucose to fatty acids, and they leave the glucose to the others. But remember, we said the muscle can store glycogen. Let's go back. We said the muscle can store glycogen, but it cannot release it. So in the first stage of, in phase two, they can also use this glycogen reserves, but again, they cannot really release it. So they use this glycogen reserves and they use fatty acids, but they also release some amino acids to support gluconeogenesis. Now, for adipose tissue, they switch from storing to releasing now. So they increase lipolysis. That's the first key part. And then they release those fatty acids for energy and the glycerol for gluconeogenesis. So they release those fatty acids for the body parts or the tissues which can use fat for energy. And they release this glycerol for the liver to using gluconeogenesis. And this overall shift helps keep glucose for brain and the RBCs. Now let's come down to the brain and the RBCs. Again, they use glucose normally because they don't have any other choice. They're also insulin dependent, so the rate of using glucose does not change throughout phase one or phase two. Okay. Now, what's happening to the LPL activity and HSL activity? Remember, we said in the beginning, insulin was dominating, and therefore insulin activity in phase one, I mean. Therefore, HSL is going to decrease, LPL is going to increase. Now it's the opposite because, as you can see, we are, for example, focus on the adipose tissue. It's increasing lipolysis. It's breaking down fats. So clearly, we're shifting from the insulin to the glucagon. And so again, what's happening to LPL activity and HSL activity? LPL activity is actually going to decrease and HSL activity is going to increase because again, we're mobilizing those fatty acids. We're not storing them anymore. And now for the last phase, phase three, starvation. The body is just trying to adapt to whatever it has. And this is where most of the questions are going to come from. Very high yield, very complicated, I should say. So please focus. In phase three, the liver purely relies on gluconeogenesis because glycogen is fully depleted. That's the first thing. Second thing is it starts ketogenesis. It produces ketone bodies. And it does that, as we said before, using fatty acids. So it's going to use fatty acids, turn them into ketone bodies. But again, even though the liver can produce ketone bodies, it cannot use them because it lacks the enzyme thiophorease. That's a question, okay? Remember it, it's probably a question. And the liver uses fatty acids instead in this stage. It also uses a little glucose, but mainly fatty acids. We have a summary table for all of the organs and what they use. So you can memorize it from later. And then muscles increase fatty acid oxidation even more. We said they use fatty acids, but they increase fatty oxidation even more. And they release more amino acids to support gluconeogenesis. So there's muscle wasting in this stage. You're gonna basically go from a buff guy to a skinny guy because you're just trying to survive. Now, even though the muscles can use some ketones, they prefer to leave most of the ketones for the brain. We're going to talk about this in a bit. And finally, or before we get into RBCs and brain, we have adipose. And adipose tissue increases lipolysis even more. So whatever we saw in phase two, it's being exacerbated times two. Think of it like that. So adipose tissue increases lipolysis even more and they release those fatty acids for energy, and the glycerol goes for gluconeogenesis at the liver. And again, we said most tissues can use fat for energy. It helps keep the glucose for the brain and the RBCs. And finally, for the RBCs and the brain, RBCs rely 100% on glucose because they lack mitochondria. It's not like they have a choice. They can't really function on fatty acids or ketones or whatever. They can only use glucose. No glucose, they're gonna die. And so the entire body, as we can see, is trying to leave this glucose for the RBCs and the brain. And now let's talk about the brain. Surprisingly, the brain now uses ketones and glucose. And so this makes sense on why the liver starts ketogenesis, because the brain can actually use ketones. So the brain shifts its focus from glucose only to ketones and glucose, and this is to leave some of the glucose for the RBCs, because the RBCs cannot function on any ketones, any thing other than glucose. And another reason you may wonder why the brain can use ketones and not fatty acids, it's because fatty acids cannot cross the blood-brain barrier, okay? So once we change those fatty acids to ketones, now the brain can use it. Now keep in mind, even though the brain uses ketones, it still uses some glucose. So if a question asks you, does the brain use glucose in phase 3? Your answer should be yes. What is the major energy source for the brain in phase 3? It's going to be ketones. Do you get the difference? Okay, now what's happening to LPL activity and HSL activity? We said that phase 2 and phase 3 are kind of similar because you're just mobilizing and releasing glucose and whatever energy source you have. So the answer is the same. This is going to be to a larger degree. So LPL activity decreases even more, HSL activity increases even more. Now, this is a very, very important summary table on what every organ uses. I'd like you to pause, read it on your own. Nothing new is mentioned over here. Just pause and read it on your own. Let's move on to question 1. Which of the following will be the source of energy for the brain during starvation? So the answer over here is D. Now the question clearly is trying to get from us what is the main source of energy during starvation. So even though we have glucose as one of the options, the main source is ketone bodies. And they might try to question you or trick you. You guys should know this by now. The acetoacetate is used in keto. It's one of the products of ketogenesis, all of that. So they might question you like, what increases in the blood in person during starvation? Acetoacetate, acetone, ketone bodies, ketogenesis. All of those are keywords, okay? They all mean the same thing. Question 2 now. Which one of the following tissues relies only on glucose for ATP production? 3, 2, 1. The answer is E. Erythrocytes is another name for RBCs. Question 3. During fasting, muscle glycogen cannot provide glucose to the blood. Which of the following is the most probable explanation? 3, 2, 1. The answer is C. Muscles lack glucose 6-phosphatase, and you just have to memorize it honestly. Okay, now for the last lecture. Bear with me, guys. But this lecture is very, very simple. I think I have like 4 slides for it only. Now, starting with the easy stuff, blood glucose. The normal levels of fasting plasma glucose is 70 to 100 milligrams. Excuse me, is 70 to 100 milligrams per deciliter. Okay? Any less than that, you're in the hypoglycemia playground. Any more than that, it's hyperglycemia. Now, if you have more than 126 milligrams per deciliter on 2 separate occasions, you're diagnosed with diabetes, okay? Type 1, type 2, that's not our concern. You're diagnosed with diabetes. And so from this, we can learn that if diabetes is not treated, you're going to have hyperglycemia. Now let's look at the HbA1c, our best friend. And when you have HbA1c level of 6.5% or higher, you're a diabetic, okay? Anything slightly lower than that is pre-diabetic, and then even less, you're normal, okay? Or hypoglycemic, depends. Don't worry about the values except the ones I've shown in this slide. Now, the best indication of increased blood glucose over weeks is HbA1c because it gives an average for the last 3 months. It doesn't give normal fasting plasma glucose gives an average for the it doesn't give an average, it gives your current status. But HbA1c basically snitches on you. It tells the doctor what's been happening the past 3 months because this is the RBC lifespan and HbA1c measures glycated hemoglobin. Okay? And lastly, high blood glucose causes the glycation of many proteins, and this causes retinopathies, which is eye problems, nephropathy, which is problems with the nephrons, kidneys basically, and neuropathy, neuron problems. Now, this is a very, very, very important. This is basically the entire lecture in one table. If you can pause, read it, and with this as well, you're done for this lecture, almost, except for one clinical case. So let's compare type 1 and type 2 diabetes. Type 1 diabetes, the patients are usually young and thin. Type 2 diabetes, the patients are usually old and obese. Type 2 diabetes is characterized by absolute insulin deficiency. Type 2 diabetes, you have insulin resistance, and in the later stages, you have insulin deficiency. Type 1 diabetes is basically autoimmune attack on the beta cells, and it's triggered by a virus. And even though there is a genetic influence, it's weaker compared to type 2 diabetes. And then the pathogenesis for type 2 diabetes is not completely understood, but they say that it's due to the increased adipose tissue, and therefore there is more inflammation, which causes insulin resistance. And then, since the cells, the muscles, the adipose are insulin resistant, the beta cells must produce more insulin. And this is going to work for some time. They're going to produce more insulin, more insulin, more insulin, and then they're going to basically uh, die or become or lose their function. Like they can't keep producing infinite insulin. So at some point, they're not going to work anymore, and this is where you start experiencing the symptoms. This is late stage type 2 diabetes, and this is where insulin deficiency happens. Okay. And then you have insulin therapy required for type 1. Always. Like type 1, you have to give them insulin. Type 2 is different. Insulin is only required in the late stage. Because as we said, in the beginning, even though there is some insulin resistance, it's being compensated by the overproduction of the beta cells. Once the beta cells begin dying, خلاص, you have to give insulin. And then presentation of type 1 is polyuria, polydipsia, and polyphagia. Let's try to understand them. Polyuria is frequent urination, and the reason this happens is because, as we said, with both the types of diabetes, you have hyperglycemia. In hyperglycemia, you have increase in blood glucose. And if you go back to renal, you can remember that glucose is like always being removed from the urine, added to the blood. And we never go over that value. Just think of it. If you have so much glucose to the point that I don't want to give you complicated names, but if you have so much glucose to the point where we cannot recover all of it, then the glucose is going to start appearing in the urine. And then one thing you should know about glucose is that it pulls water with it. So quick recap, glucose is increased so much, it spills into the urine, it pulls some water with it, and then you have frequent urination. This links us to the second symptom, which is polydipsia, which is thirst. Basically, you have to drink, you're always thirsty, you always want to drink water. And this makes sense because you're losing so much water in your urine. So polyuria, polydipsia, they're both linked together. And then polyphagia is because you don't have insulin, you don't your body is not building muscles, your body is not building fat. You're always in this stage of glucagon and releasing, and you need energy, so you're gonna be hungry. And as for type 2 diabetes, it's usually detected by routine screening. However, in extreme cases, when the beta cells die, you can present with some of those, okay? Now, some key factors for type 1 compared to type 2 is that there is no hyperinsulinemia. You don't have increased insulin. Actually, you don't have any insulin. And you also have ketoacidosis because, as we said, there's no insulin, and therefore you have glucagon. Glucagon, as we mentioned before, in phase 3, results in ketogenesis, and this increased ketones in the blood is gonna make the patients present with ketoacidosis, okay? And lastly, we have hyperlipidemia in type 1. Hyperlipidemia is not present in type 2. So, quick recap on this last one because you can answer so many questions if you just memorize this. No hyperlipidemia, ketoacidosis, no hyperinsulinemia, ketoacidosis, and hyperlipidemia are found in type 1. Type 2, there is no ketoacidosis, you have hyperinsulinemia, and there's no hyperlipidemia. That is 90% of metabolism in diabetes. Now, there's one thing which I really want to talk to you about, especially since we're fasting now, which is diabetic hypoglycemic attack. Usually, when you're fasting and then you eat a meal, the insulin overshoots, okay? And this is normal. It's known as postprandial or reactive hypoglycemia. Insulin overshoots, and then the glucagon is going to fix it back, okay? So the levels go back to normal. This is in a normal individual. But in a type 1 diabetic specifically, this doesn't happen. So the patient would usually present with them taking their insulin shots or insulin IV injections, but they forget to eat. So think about it. They get insulin, exogenously, of course. They forget to eat. The body now thinks that, oh, we have insulin. It means we have so much glucose. Let's absorb all of the glucose. And then the patients would present with severe hypoglycemia. Now, normally glucagon would compensate, but in individuals with long-standing type 1 diabetes, the alpha cells which produce glucagon are also dead. So you don't have glucagon, you don't have insulin. With so much insulin just not being opposed, you're gonna have severe hypoglycemia and it's a medical emergency. Okay? Now let's talk about the treatment of type 1 and type 2. Type 1 has no treatment. The standard care is giving insulin. Okay? Just memorize this. Type 2, for newly diagnosed patients, you can save them by decreasing body weight because decreasing body weight has been shown to improve glucose tolerance and insulin sensitivity was the word resistance. So it's gonna reduce the insulin resistance and improve glucose tolerance. And exercise and dietary modifications also are important for newly diagnosed patients. As for late diagnosed patients, it's not going to work as much. It's still beneficial, but you still need medications. Now, some anti-diabetic agents for type 2. Remember, we said type 1, just give them insulin. Type 2, you can give them two drugs, which are metformin and sulfonylureas. Metformin works by decreasing the glucose production overall. This is the overall function. And it does that by inhibiting gluconeogenesis in the liver. Okay? So if you were to memorize one line for metformin, it's going to be this one. Inhibit gluconeogenesis and therefore decrease glucose production. And the other one is sulfonylureas, and they work in a different way. They basically, they're over, like the end stage or the end result they reach is increasing the insulin secretion. And they do that by blocking the ATP sensitive potassium channel, also known as KATP. And then this results in depolarization of the beta cells. When they're depolarized, you guys should know this by now, the end of the nerve, the last stage of depolarization, calcium moves in, and then calcium moves in, causes the vesicles to fuse with the plasma membrane, release the insulin. Okay? So quick recap, sulfonylureas sulfonylureases increase insulin secretion, and they do that by blocking KATP, which depolarizes the beta cells and releases insulin into the blood. And lastly, let's talk about gestational diabetes. Again, I told you guys before that the vibe I'm getting from your exam is clinical oriented. So try to focus on your clinical aspects of your lectures. Gestational diabetes. So this is when the mother has diabetes when she's basically pregnant. And it doesn't have to be like type 1, type 2. لا. In some cases, they develop diabetes while they're pregnant. Basically, increase blood glucose because they're trying to feed their babies. And then, in some cases, even after the delivery, it remains and the mothers become diabetic. That's not our concern over here. Just remember, gestational diabetes has severe consequences. And a consequence is fetal macrosomia, which is a large baby, as we can see over here, mashallah. And they're also shaky. The reason they're large is due to the anabolic effects of insulin. As we said, insulin causes the storage and the production, storage and the building of stuff. So the baby over here is clearly large. This is due to the anabolic effects of insulin. And the baby is also going to be shaky. They're going to be hypoglycemic. They're going to shake, okay? And this is purely due to the, honestly, you just have to memorize this. It's due to the baby's relative hyperinsulinemia. So since the mother has a very high blood glucose level, the baby is also going to produce a lot of insulin to counter that. So once the baby has been delivered, the baby is going to experience hypoglycemia. And this is due to the baby's relative hyperinsulinemia. And question time. Other than obesity and genetic profile, what is the most important thing in telling that it is type 2 diabetes? 3, 2, 1. Remember, we said that type 2 diabetes is classified by insulin resistance and, in later stages, insulin deficiency. So, other than obesity and genetic profile, the answer is going to be muscle resistance to insulin. The answer is E. Question 2. What is the function of metformin in a diabetic patient? The answer is A. Decreases gluconeogenesis by the liver. You just have to memorize it, honestly. Nothing much. You know what? Okay, let me give you a question on this. What is the function of sulfonyl ureases in a diabetic patient? If this was an SAQ, what would you write? Your answer should be it inhibits the potassium ATP channels, and then this is going to depolarize the beta cells, and this is going to result in calcium entry, vesicle fusion with the plasma membrane, release of insulin, and that is it. Increased insulin production overall. Question 3. A 12-year-old boy presented with fatigue, polydipsia, polyuria, and polyphagia. A finger stick glucose measurement shows a glucose level of 350 milligrams per deciliter in his serum. He is diagnosed with type 1 diabetes. Which one of the following is most likely occurring in this patient? It's pretty complicated. I'll give you 5 seconds. You can pause, of course. 5, 4, 3, 2, 1. Okay, let's go through it. Type 1 diabetes. So, will there be a decrease in hepatic gluconeogenesis? Not necessarily. You don't have any insulin, so it's going to be the opposite. You're going to have increase in gluconeogenesis. Then B is insulin resistance. No, we said type 1. There is no insulin resistance. You don't have any insulin to begin with. And then C is decreased conversion of fatty acids to ketone bodies. Not really. There's an increased conversion of fatty acids to ketone bodies, and that's why we have ketoacidosis in type 1 diabetes patients. And then D, increased production of acetoacetate. And remember, this is how they like to trick you. It is correct. Acetoacetate, ketone bodies, ketones, acetone, ketoacidosis. They're all the same, okay? And then lastly, increase stores of triacylglycerol in adipose tissue. You're not storing, you're releasing, you don't have any insulin, okay? This is the answer key. Try to solve the questions without looking at the answers. This is the only reason I have them at the end. I know it's not very convenient, but it's a review. You guys should aim to answer all of the questions. And if you've watched till the end, thank you very much. That is all. I wish you guys the best of luck and just try to push through this burnout season. Don't underestimate MOL, but don't sleep on MSK as well. If you have any questions, since I had to basically study for your midterm again, feel free to ask me, even if it's a low yield point, feel free to ask me. And thank you very much.